Good afternoon, everyone. Thanks for coming to this session 11, Offspring and Next Generation. I'm Prakash Hande from the C1 member of ICRP, and also I'm co-chair of Task Group 121 on Offsprings and effects. And I'm from National University of Singapore. And my co-chair is Dr. Yoshia Shimada from IES. He's also a member of C1 and the task group. Yeah. Okay. So this session is on Offspring and Next Generations. We have 5 speakers on a variety of topics which deals with the radiation effects on embryos and offsprings. So we will have the first presentation by our rapporteur, Dr. Hisanori Fukunaga, who will introduce some of the aspects of the speakers as well as the posters submitted to this particular session. Hisanori, please.
Thank you. Please, a moment. Hello, everyone. I'm Hisanori Fukunaga from Hokkaido University and serving as a rapporteur of this session. I would like to briefly introduce the poster presentations related to this session. There was no pre-recorded video presentation. Four relevant posters have been accepted for this session. All of them were from Japanese research groups. Unfortunately, this session seemed to have fewer submissions than the other sessions, since approximately 250 presentations have been scheduled for this symposium. Okay? And this slide shows all accepted posters for this session. The first poster entitled the emerging data that revolutionizes the fundamentals of radiation protection over 100 years and the theoretical reproduction and interpretation by the WAM model is presented by Dr. Bando from Kyoto University. The second poster is presented by Dr. Toki from Osaka University, and the third poster is presented by Dr. Tanaka from IES, and the last poster is presented by Dr. Mirai from Tokyo Institute of Technology. Okay. I have categorized these presentations into 3 groups, namely Lodo's mouse experiment 1 and 2, and Lodo's experiment in vitro. 2 of the accepted posters are related to the research of Dr. Gondo, who will speak in this session. Dr. Gondo and his colleagues conducted whole genome sequencing experiments in mice under Lodo's radiation environment. For more details, please listen to his presentation. The 2 posters can be considered completely complementary to the research in terms of radiation protection and statistics. The third poster reports the late effects of low-dose gamma rays on the offspring of wild-type male mice. The presenter is Dr. Tanaka, right? Please ask her for more details later. Interestingly, the results show significant lifespan shortening in F1 male mice exposed to 201 milligrams per day, but no significant effect was observed in F2 mice. And the last poster reports the low-dose late radiation-induced mutations in cultured human cells using whole genome sequencing. Normal human fibroblast NB1 RGB was seeded as a single cell in 96 well plates and grown under irradiation with cesium-137 gram rates of 1 or 20 milligrams per day for 3 weeks. They found lots of mutations, but there was no significant difference in the number of mutations between the non-irradiated and irradiated cultures. They also reported a predominance of C2A transversion associated with guanine oxidation. Okay, so all 4 posters report significant results in this area. If you are interested, please go and have a look. Thank you for your attention. Right, thank you.
Share the first 2 or? Yeah. Okay. So we have next presentation by Professor Richard Wakeford, who is from University of Manchester. He's also a member of C1 committee, ICRP, and co-chair of Task Group 121 on Offsprings. So he's going to update us on ICRP Task Group 121. Richard, please.
Thank you, Prakash. So you will see that I'm going to run through just what the basics of Task Group 121, and that this is a presentation by me and my co-chair, Prakash Hande. Here's the full membership of Task Group 121. Quite a lot of different expertise there being brought to the table. So it leads to a lot of interesting discussions. Now, just to emphasize that we're dealing with both preconceptional and intrauterine exposure, but
Can they hear?
you must understand that these are very different things. So if we look at preconception exposure, of course, we're considering the effects of irradiation of the parental germ cells, hereditary effects of exposure. But once we get past conception and we deal with the developing organism in utero, we're dealing with somewhat different effects, developmental effects as the organism develops in utero, teratogenic effects, carcinogenic effects, cancer in childhood and later life, and of course the irradiation of the fetal gonads as well. So we are dealing with these two different aspects of risks in offspring and next generations. The risk of hereditary effects was considered really last comprehensively, anyway, in the UNSCARE 2001 report. And that flowed into the ICRP publication 103. A lot of this was due to the late Professor Sankaranarayanan, who published a considerable number of papers contributing both to the UNSCARE and ICRP publications. But I've indicated down here, taking this table 1 from ICRP publication 103, how the, when it says present, that means ICRP publication 103, and how that differs from publication 60 in 1990 recommendations. And you will see that there's quite a difference in the risk estimates, the detriment, at least, from heritable effects, more so than for cancer. And so that is an interesting aspect, which we are going to be examining. We know basically at least some of the reasons why these risk estimates have reduced, but we are going to look at these in some detail. And of course, there are still no reliable risk estimates for hereditary disease from human epidemiological studies. So these risk coefficients have to be based on large experiments, mainly mouse experiments of laboratory animals, and a knowledge of human genetics. So we're going to go into some detail here to answer the question: have the assumptions underlying the risk coefficients for hereditary disease changed since those made for publication 103, and if so, by how much? So that's our basic remit for hereditary effects. As far as intrauterine exposure is concerned, as I pointed out in that summary slide, the in utero exposure can lead to an increased risk of teratogenic effects, congenital malformations, severe retinal retardation, These are treated as tissue reactions at the moment. There are questions over reduction of IQ. Is there a threshold for that? There certainly is for severe mental retardation. And we do have, of course, epidemiological evidence from the atomic bomb survivors irradiated in utero for severe mental retardation. So we'll be having another look at that. Then we have carcinogenic effects. Cancer in childhood, these are the big case control studies of childhood cancer and how that relates to antenatal exposure. Of course, we're going back to the initial results that are almost as old as me from 1956. That first indicated an increased risk of cancer in childhood following exposure of the pregnant mother to diagnostic radiation, and of course in later life, and much of that evidence for cancer in later life comes from the atomic bomb survivors irradiated in utero, a recent paper from Sugiyama et al. But of course, at the time of that study was published, only 15% of those survivors were actually died. So there's quite a lot of information to come. And there are indications of the risk of cancer extending into adult life, but in women rather than men, strangely enough. But so again, there's something to be sorted out there. And finally, of course, we have hereditary effects, which is due to exposure of the gonads of the fetus. So some overlap with what we're looking at in terms of hereditary effects anyway. So last time ICRP looked at this comprehensively was in publication 90, which is in 2003, but it hasn't been considered in detail since. But there has been some more evidence coming in which you're going to have to consider. So we're going to be looking at this additional evidence and what we're going to say about the risks of in utero exposure. And also, we're considering non-human biota, which hasn't really been considered before, and certainly in the system of radiological protection. So we are going to be looking at filling the gap of hereditary effects arising from irradiation of non-human biota. So that will be part of the task group's work. And of course, it's the effect of, as I'm sure you all know, its effects at the population level for non-human biota, and also for the implications for the system of radiological protection. This will be a change from publication 103. So, the task group 121 has this remit that covers wide fields of hereditary effects arising from preconceptional irradiation, parental gonads, and of the teratogenic, carcinogenic, and hereditary effects arising from irradiation of the developing organism in utero. And both human and non-human species will be considered by the task group. The impact on the current system of radiological protection and what that might mean for the next set of recommendations will be assessed by the task group. Okay, thank you very much. That's me done.
Thank you, Richard. We have a question and answer session at, when all the speakers have presented their talks. So we'll go for the question and answer later. So we have now a second speaker for this session. So we know that in the past we used to measure cytogenetic parameters, morphological parameters after in utero exposures in mice. But with the advent of newer technologies, we have now whole genome sequencing, and we can generate a lot of data on the effects of radiation based on in utero exposure. So Dr. Yoichi Gondo from Tokai University School of Medicine is going to tell us about the challenges to assess the next generation risks using whole genome sequencing in a mouse model. Dr.
Thank you, Chairman. Yeah. It's a privilege to be here, and thank you, Dr. Fukunaga, about the kind introduction. So I can get into this low dose rate immediately. So this is a system to give them a low dose for a long time, up to 400 days. Dr. Nori Nakamura mentioned just last session, it's tedious to do a long exposure with the low dose, but we did it. One is this low, 0.05. Dr. Shimada used to say, always says, this is about a double dose of the background level, up to 20 mg per day. So two different institutions. One is IES, Aomori Prefecture, and one is at the Tokai University, where I belong, at the Kanagawa Prefecture, about 100 kilometers away, totally independent institution. I'll give you a punchline first, or a punch slide. This is a conclusion, actually, well, result, I would say. So by 20 mg per day for 140 days, showed a significant difference from the control with this statistical value. How we get it? The basic method is trio analysis. Dr. Eager conducted in a large-scale human cohort, Chernobyl. But we did it in a mouse model because we can set up exact control system under the same condition. Just the difference is the radiation, all right? And the principle of the detection of de novo or newly arisen mutation is this. So assuming on average, either oocyte or sperm gives M number of M new mutations, then subtracting the pre-existing mutation in their parents, we get 2M. This is a mutation rate per mouse, per generation. Remember, 2M is the mutation rate per generation per mouse. Then, to enhance the detection, we conducted multi-generational exposure, starting from the same pair of the mice, then up to 4 generations. Then every generation accumulated 2M, 2M, 2M. So in total, at the G4, we get 8M. So from this number, if we like to estimate the mutation rate per mouse per generation, simply we can divide it by 4. So this is a scheme. So exposure started just at the time of the G1 mating. Control, no irradiation. All right, so if any effect, we could expect additional number of mutations from G2. No effect on G1, but from G2. So there's a 1 generation gap, lag. Then we have to whole genome sequence only, of course, we have to do parents and only G4s. We don't have to do these intermediates. Well, to make it simplify, we forget about X chromosome because this scheme only applies to the autosomes. So today's talk, I focus on only autosomal mutations and single base substitutions. All right, this is a Tokai result. Starting from the 1 pair of G0, male and females, then we get 9 each for control and exposed. These are the number of detected, practically detected de novo mutations which didn't existed in parents, or actually G0 ancestor. So let's plot, well, per mouse per generation mutation rate, we simply divided by 4. So these are two M's, if any, plus some R. Let's plot it back to the punch slides. Here is the Tokai result, average. This is the whole result of 9 control and 9 exposed. Then Institute of Environmental Sciences, IES, conducted much bigger scale, 0.05, 1, and 20 mg per day for up to 360 days, 1 year. Starting from last year, 2 years ago. Then finished 1 year later. This is the result. Well, I'll come up to 20 mg later. First, I focus on the 0.05 and 1. This is the result divided by 4 and plotted back to the slide. Then this triangle, no significant difference at all. So we didn't see any radiation effect up to 1 mg times about a year, all right? Then what about 20 mg? First of all the females at G2 became infertile. So we couldn't get any G3 at all. So we modified, mating this male, fortunately they are fertile, to one of the G2 control female. Fortunately, we got offsprings. And at the same time, July 20th, year 2022, we get 6 siblings from each pair, control and exposed. And we get this number divided by 4, gives us 2M, if any, plus some R. Plotted back to the plot. This is the first punch slide. Remember? So significantly different. First, I'd like you to focus on these 3 controls exhibiting very reproducible system of our whole genome sequencing method or expanded TRIO method. Anyway, it is significantly different statistically. This is the first time, I think, as far as I know, the detection of low dose rate 20 mg for about actually exactly 40 days gives us significant induction of gamma ray induced mutations. This is the lowest, I think, so far. So now we can focus on this area. I'm very curious, probably you too. Anyway, I keep talking about number of mutations per mouse, but you might be interested in mutation rate. Then you need what a part of the genome this system covered. Effective whole genome coverage, or EWC, is the number, what a part of the genome we covered. Out of 3 gigabase mouse genome, actually, we disregarded X chromosomes. So only autosomes, so a little less, about 2.5 megabase haploid genome, we covered about 2 megabases consistently. And divided by number of per mouse per generation, divided by EWC gives a mutation rate. Very simple. So sampling size was summation of this number of mice is 42, I've been saying, but practically, real sampling size is this. We looked at this number, 168 gigabase pairs of base pairs of mouse genomes. Then we found a total of about 4,000 de novo mutation. This is the size of our mutagenesis study. That's why gave such a reproducible result. Well, I have 1 minute or so, probably. So I explained up to here. And this one, all data deposited to DDBJ or GenBank, EMBO, SRA, DRA is a platform. So you can use freely soon. And also, we like to open to public this all the mice in the Tokai University and most of the mice at IES, frozen, whole body is frozen stock. So if you are interested in structural variations, reversion, inversion, we can do pack biosequencing using intact long DNA from this frozen stock. So, well, there's a question to you. This is a question to you. And I may leave this slide for the discussion time because time's out. Thank you.
Thank you. Thank you, Dr. Gondo. Very interesting data generated from this low-dose rate exposures in mice, especially, removing the X chromosome data for the analysis. Yeah.
Hey, so we'll move on to the next speaker, and Dr. Asao Noda. As you know, the genetic effect in humans are heavily based on the results from A-bomb survivors, and he will show us many data regarding with the genetic effect among A-bomb survivors. Please, Noda.
So Dr. Gondo's data is so intensive. So my talk is something very old study. So thank you very much for the chairman and the ICRP committee to give me an opportunity to talk about RERF study program regarding the genetic effect of atomic bomb radiation exposure. Sorry. Studies on atomic bomb radiation studies on radiation genetic effects on the offspring of A-bomb survivors is one of the oldest studies in RERF, and it's been continued more than 70 years. And now, genomic era, we are planning whole genome sequencing of the parent-child trios. So I'm going to follow the history of the study. The question is whether we can detect the germline mutation on the offspring of the A-bomb survivors. However, there are some limitations to detect the effects. Firstly, offspring has been born alive. Infertility or early embryonic deaths are difficult to assess. Say, the large deletion or chromosomal structure changes may be eliminated in the reproductive process. And also, the genomic or epigenetic changes need to be visible in the F1 phenotype. Say that typical dominant mutations are easy to detect, but recessive mutations are invisible. And also, mutations in the non-coding regions are difficult to assess the biological implication. And finally, we don't know how many haploinsufficient genes exist and the degree of their influence in humans. This is a whole picture of RERF genetic study program. In early days after the bombing, that's from 1984 to 1958 to 1954, RERF investigated the birth defects such as stillbirth or neonatal death. And then F1 cohort consisting of 77,000 F1 established and continued to collect the data of F1 cancer incidence and F1 cancer and non-cancer mortality. In parallel, 12,000 F1s are selected for the F1 clinical studies and continued their physical health examination. Laboratory genetic studies are separately initiated. In early days, 16,000 children are selected for the examination of chromosome abnormalities. Then the mutation assays expanded to chromosome fish and classical genomic DNA analysis by gel electrophoresis and array CGH And then specific locus tests, such as mini-satellite, microsatellite changes. And now whole genome sequence error. So I will review the past result briefly. Birth defects. Neonatal diagnosis was done at that time along with interview of the parent exposure status, such as birth weight or any defects such as stillbirth, malformation, and perinatal death, sex ratio, etc. The conclusion was that there was no evidence was obtained that which indicate the effect of parental exposure to radiation. We recently reappraised the old data by using the oldest, newest DSO2 those dosimetry systems and reconstruct the individual data to minimize any biases, and look at within 7 days or 14 days after the birth. The results showed that as a parental dose increases, congenital malformation and perinatal death tend to increase, but it didn't exceed the statistical significance. However, there are still a small correlation between the parental total dose and the perinatal death in 14 days. We conclude that these small changes may not be due to solely radiation, because the parents who received higher dose were closer to the hypocenter. They had experienced severe living condition to survive. That could be a considerable burden on the mother and the newborns. This is the history of the study. And the recent report, Dr. Yamada concluded that future genome study will provide the opportunity to more precisely discuss the genetic effect of the radiation, parental radiation. Here are the mortality and cancer incidence studies that has been followed up in ICRP epidemiological studies. The data are a little bit old, but totally 75,000 subjects, and the average parental dose was 264 milligrams. And also, there is no evidence of the increased cancer incidence in the offspring of irradiated parents. So, DNA study began from around 1995. ICRP has been collecting the blood sample from the member of the about 14,000 families, trios. And until now, classical genomic DNA study in ICRP didn't show any evidence of the parental exposure on the children. So, why we cannot detect mutation in the radiation-induced mutation in survival offspring? We know the mutation rate per locus is quite low, and there's no versatile genetic markers in humans. And animal study at very high, quite high dose, and it's rare such a high dose in human cases. Now, the genomic error, several reports of parent-child trio whole genome sequence is already reported. This is, one of the famous papers that they mentioned the number of de novo mutation increased with the age of the parents, especially in the father age. And also, they reported the individual genome difference is considerably different between individuals. So in such a condition, we need to look for find the radiation-induced mutation in the offspring genome. Here the animal study, just Dr. Gondo mentioned that one study from the UK, they reported small indel and multi-site mutation induced after the paternal sperm stage exposure to 3-gray. Also, our RERF study also indicated small indel and multi-site mutation, the induction of the mutation at paternal spermatogonia or maternal mature oocyte stages. So, if we can fully analyze the genome data, it is possible to find the radiation-associated mutation in the offspring if there is a statistically sufficient number of high-dose parents. As you know, the Chernobyl studies, Dr. Yegas here, their report indicated that they didn't observe the radiation-induced mutation in the offspring of the Chernobyl. They only observed the age effect. So the RERF study, we now realize the difficulty in detecting new mutation in the offspring. However, if we fully characterize the radiation-induced germline mutation, we may be able to discuss radiation-associated genetic risk, is there a risk or not. So the RERF study plan that maybe Dr. Uchura might mention later, the whole genome scan with a SNP chip, and then short read and long read sequencing with high coverage to detect radiation-induced mutations. Anyway, ethical issues are particularly important in human genome study. Even more, Torio whole genome sequencing has some aspect of victim research. So, RERF has been working on the LC issue and making a public awareness campaign to communicate the representative of the participants and also citizens. Of course, our efforts of course, follow the Japanese ethical guideline for human genome study, but even more because this is a victim research. And ultimately, the study needs to be beneficial to the participants in the long run. So, RERF has held an LC workshop on the Torio genome study. RERF held a repeated meeting with the second generation atomic bomb survivor organization or unions for more than 2 years. And RERF has held a stakeholder conference to develop a comprehensive informed consent and community understanding. These are one of our efforts for the communications. Stakeholder meeting has been going on over the years, and they supplied us important issues for the studies. The major comments are that the study need to respond to the concern of the participants and the citizen, and that the study need to be transparent to the participant and or the society, and that study should not be used for the military purposes. So there are still concerns and anxieties we are being asked the participant to carry out the study as soon as possible, because the survivors are parents. They are elderly. They don't have much time left before they know the results. So this is my last slide. Recommendation from the stakeholder committee. There are several strive to foster the trust in RERF. And they request to do the whole genome sequence as soon as possible. And they need, it is necessary to explain the significance and interpretation of the result to society, and give a sufficient consideration for the disclosure of the genome data, and also give the sufficient explanation in the consent form regarding the return of result to individuals. So we plan to continue to communicate the participant and the citizens even after the initiation of the study. Thank you very much for your attention.
Thank you, Noda, to show us a very comprehensive study results. Okay, we would like to move on to the next speaker, Dr. Arikuni Uchimura, and he will talk on the whole genome sequencing, which will be applied to the ABOM survivatory study in future. Please.
Okay, thank you, Dr. Shimada, and thank you for this opportunity, great opportunity for giving my presentation. I'm Arikuni Uchimura, researcher at the RERF. Okay. As Dr. Noda said, RERF is now planning to conduct whole genome sequencing study family trio, including atomic bomb survivors and their children, to determine the genetic effect of radiation. And this is RERF so far have collected the blood sample totally 1,500 of F1 children. And this blue colors number is children, the number of children born to the father exposed to 1 gram or more. And these 116 children are born to the mother exposed to 1 gram or more. So these totally, our current plan, we conducted 219 F1 from high dose group and the same number of unexposed or zero, close to zero exposed control group. But today I'd like to talk about mainly our current study, current result, and mouse animal model study. First of all, I'd like to explain our mutation detection methodology by using short read next generation sequencer. And this method was also used in the Dr. Gondo's analysis, the low dose rate experiment. And I mentioned very briefly, we conducted whole genome sequencing by short read NGS with more than 30 coverage. And the obtaining read data are mapped to the mouse reference by BWA software. And for detecting small size mutation, we use haplotype caller to detect mutation, and we focus the mutation in the effective whole genome coverage region. This region is common to the exposed sample and control sample to compare precisely. And for detecting structural variant, we use several softwares, for example, such as MANTA, SVABA, and RAFAS, and assess the entire genome region. And this is a result, and this result partly introduced by Dr. Noda, but I explained much in detail, more detail. And we conducted the two types of experiment. One is a paternal exposure experiment. at the stage of spermatogonian stage exposure and with 4 gray gamma ray. And the other case is a maternal exposure case. This is a mature oocyte stage, 4 gray gamma ray exposed to like this. And we tested unexposed parents, children 6, and 6 mice from the such irradiated mouse F1. And we compared de novo mutation and single nucleotide variants and insertion and deletion and multi-site mutation. Very clearly increased the insertion, deletion, and multi-site mutation in paternally exposed model and also maternally exposed model. And in addition, we also analyze the structural variants in these families. In this case, is a maternal exposure model. But control is a three de novo structural variants, but exposure case is a 5. So we could not detect a clear statistically significant difference in this experiment. And Dr. Noda explains that UK Adeboye and his colleague reported the transgeneration effect study result. And their experiment is a 3-gray X-ray exposed to the sperm stage and investigates children and the number of mutation. The single nucleotide variant is not changed, but indels and the multi-site is also increased in the paternal exposure case. So these results suggest such indel and multi-site mutation. Multi-site mutation is a multi-locus is simultaneously altered this type of mutation. And this type of mutation, the specific signature holds radiation-induced mutation, and also suggested that the time of exposure is not so important to increase this induced mutation. And the next topic is radiation-induced mutation in mouse somatic cells. This work was conducted by my collaborators, Dr. Matsuda and Dr. Tanabe. They exposed mouse at approximately 8 weeks. And 8 weeks later, they collected the bone marrow cell and isolated single long-term hematopoietic stem cell. And cloning each cloning cells are expanded in vitro and subject to whole genome sequencing analysis and to detect somatic mutation in each single cell. The result is very clear. Single nucleotide variants, no exposure, and 3.8 gray and 7.5 gray increase the SNV, the number of SNV, in a dose-dependent manner. And small insertion and small deletion is also the similar increasing tendency. And the non-repeat deletion is a very specifically detected only in the exposed population. So this is a very distinct signature of radiation-induced mutation. And they also observed the multi-site mutation detected only in such high-dose exposed hematopoietic stem cell, and structural variants increased in a dose-dependent manner. So this is a summary of the such radiation exposure experiment. For mouse germline, 2 indels and 0.8 multi-site mutation were induced per 1 gray exposure. In contrast, mouse hematopoietic stem cells, 15 SNVs and 15 indels and 1 multi-site mutation and 0.8 structural variants were induced 1 per gray per diploid genome. Assuming haploid genome, these numbers are halves. The indels and multi-site mutations appeared in both experiments. But the number is higher, much more in the HSC. So these data suggests germ cells, mouse germ cell is more resistant to radiation than the hematopoietic stem cells. Last, last topic I'd like to talk about the spontaneous mutation in mouse germline. This is my life work study. We are conducting a mutation accumulation experiment using laboratory mouse. This experiment is very simple. The repeated sister and brother breeding for more than 17 years, and we compare the first ancestor and the descendants' genomic information and phenotypic change. And this is a family tree of our mutation accumulation experiment. We initiated this study, this breeding. First breeding started in 2006, so 17 years we have continuously bred many mouse breeding. And we using the wild type BRAC6 mouse, and we also using mutator mouse. Mutator mouse is a spontaneous mutation rate is increased mouse. This mouse is having the DNA polymerase delta modification. This resulting in the spontaneous mutation rate is 17-fold higher. We reported the result of this experiment firstly in 2015. Our results suggest that mouse per generation spontaneous mouse mutation rate is about half of the human rate, and our mutated mouse is a 70-fold higher mutation rate. This phenotypic results, I think this is a very important data. And the higher 70-fold higher mutation rate mouse that we call mutated mouse. Mutated mouse is a breeding mutated mouse very frequently appears abnormal phenotype, like a dilution coat color and cataract, and very dim, and many type of including many disease-like phenotype, and also decrease the body weight in mutated mouse breeding in the course of generation, and dramatically decrease of the reproductive capacity in the such higher mutation rate mice. And now we are trying to detect much more detailed de novo mutation in genome-wide scale, and we using short-read NGS and also use the long-read NGS techniques. We assess the four strains and detect the de novo mutation, spontaneous mutation. This is a wild-type mouse experiment. And the results show the structural variance occurrence frequency is much higher than our previous estimates. And our data suggested about 30% of structural variant could be detected only in long-read NGS data. This is one example of the complex mutation, which is detected only in long-read NGS. For example, this 200 kilo base region are duplicated, and part of them are inverted. This type of mutation, but the long-read sequencing technique, we can easily to assess this type of mutation. This is the last slide, my last slide. Mutation accumulation experiment, higher mutation rates have deleterious phenotypic effect in descendants. And also, the latest sequencing technique enable us to detect further complex mutations. So, this is conclusion in study of atomic bomb survivors trio. We would like to advance our understanding of genetic effect of radiation by using the most up-to-date technology possible. And so, this experiment is very important, I think. Thank you very much.
Thank you, Dr. Uchimura. We are expecting that your method will, make it clear to the mutation among trio study. Okay.
So we have the last presentation by Dr. Meredith Yeager from NIH, USA. So we have, one of the cohorts we have is from atom bomb survivors, and the second one is from Chernobyl evacuees and workers, cleanup workers, for example. And Yeager's group has done some molecular epidemiology study in this cohort, and she's going to show us about the effects, transgenerational effects of radiation in cleanup workers and evacuees of the Chernobyl accident. Yeager, please.
Yeah, thank you so much for inviting me to give a presentation about our work from the National Cancer Institute. So I think we've heard a lot about how radiation causes DNA damage in cells, and I think that that's pretty clear, especially from some of the talks we've heard even in this session on the mice models. Our specific question had to do with whether or not gametes could be exposed, and if whether mutations could happen in gametes. And the way that this might occur would be in spermatogenesis, for instance. In this particular lineage, let's say a de novo mutation happened, maybe caused by radiation, and so a sperm would be passed on to an offspring, in this case with a higher mutational load, for instance. And we've also just heard about some really nice mouse model work where this has been shown to be true, both transgenerationally and also somatically. So our study was conducted on the liquidators that were called in after the accident happened in the Chernobyl nuclear power plant in Ukraine on April 26, 1986. So the resulting open-air reactor core fire was contained within about 9 days, but people from the surrounding area were enlisted to come in and clean up after the accident, some of whom were not informed of what exposure they were being subjected to. And so our study question specifically was on these individuals, these liquidators, and the question is there a transgenerational effect of radiation following the Chernobyl accident? In other words, is anything about the parental exposure passed on to their offspring? And so it would be this situation that I described a few minutes ago. Okay, so a field study was conducted by recruiting the liquidators and some of their children from 2014 to 2018, which was published, the study design was published here a few years ago. And we did this in partnership with the NCRCM in Kyiv. Families were recruited based on exposure categories, and careful dosimetry reconstruction was done, since all of this had been done after the fact, and all of this was done through questionnaires, extensive questionnaires and interviews with the subjects. And so I'm not going to go through all of the different combinations, but we did have all the different combinations of whether the father or the mother was exposed or unexposed, whether both were. And then we did have a number of individuals who experienced radiation sickness, only a small number of those, and those are mostly the first responders. And so, similar to what my colleagues have described in this session, we performed high coverage, or ADX whole genome sequencing. This was short read technology, and we had 105 mother-father pairs and 130 adult children. So there were some families in which there were multiple siblings. All of the children were conceived after parental exposure, and so they were born between 1987 and 2002. And all of this work was done around the time that some of the first de novo mutation whole genome human studies were being published, and so we adapted our filtering strategy based on several published approaches. And in the end, one analyst, namely me, manually inspected all the putative events because this was a very important question. We needed to get it right. And so just as a refresher, because we've heard about this already, de novo mutations. So every generation, a small number of variants are generated due to random mutations in DNA of gametes. So this is normal. This happens every generation. And these are the variants that fuel evolution. So we must have them. So the ones that are transmitted are known as de novo mutations. And we can only do these types of studies if you actually have the full set of individuals, including a father, a mother, and at least one child. And these de novo mutations are only observed in the children and not the parents. And so here's a little cartoon that shows, in this case, you've got the father's chromosomes, the mother's chromosomes, and say this black mutation or variant is in the mother, and it happens to be transferred to her daughter, and you can see that, so that's an inherited variant. In contrast, a de novo mutation is where you have, in this particular light blue father's chromosome, a mutation that arises in the child that was not present in the father, and so therefore it manifests itself as a Mendelian inconsistency and is a putative de novo mutation. I'm not going to go through this, and we are actually currently refiguring our pipeline, because this is a pipeline that we developed about 2016, I'd say. But just to say that there was a custom pipeline that we put together to do this particular study that was based on, like I mentioned, other studies that had been published. We had evaluated 17 different pipelines, and we modified basically 2 of them around 2017. And as I mentioned, we all of the putative de novo mutations that made it through the pipeline were actually manually inspected by me. And so first of all, we just looked at the total number of de novo mutations and calculated the mean, median, range, and standard deviation for the entire 130 child set. I should note that this total number, or the average number of total de novo mutations, is slightly higher than had been previously published, but we believe that that's probably because we did not exclude any repetitive areas of the genome, and also we had very high depth coverage. We thought it was very important to break the different de novo mutations into different categories, mostly because the mutations in these different categories are either arise by various different mechanisms and also are repaired by other different mechanisms. And so the highest number of class of de novo mutation was single nucleotide variants. And then we also broke, oops, I'm going backwards. We also broke out the number of indels, and so insertion deletion polymorphisms, where we had a subset where we tried to, because of previous literature, in humans and in mice and other organisms, we tried to look at the number of microsatellites, but because of the short read technology, there may have been some limitations here. And then we also, these are the multi-site variants that were previously mentioned. We broke those out because they would have a different mechanism of occurrence, and then complex variants are these rare things that looked a lot like gene conversion events or something that very complicated happened. We were able to assign parent of origin to about 40% of the data set. Mostly the variants were phased to the paternal haplotype, which I'll show you why that was true in just a minute. And so conveniently, we had a positive control. As mentioned previously, there is a very well-established parental age effect. So in this case, for both of these graphs, the age at conception for the father and for the mother is on the x-axis, and on the y-axis the total number of de novo mutations. And you can see that there's a strong positive correlation, mostly with the father parent of origin, but also for the mother as well. And so this was a convenient positive control for us because we were able to observe this effect. We knew that our data were reliable. On the other hand, when we changed the x-axis to paternal dose or maternal dose, you can see that there is no effect that we could observe. And in fact, if you look at the actual table of the estimates and the 95% confidence intervals, you can see here that we found the very, very strong father's age effect, somewhat weaker, but also already reported mother's age effect, but no effect with either maternal or paternal dose. And I don't have time to go through all the results, but we also looked at all the different classes that I described, and we also did not find any association with dose or any of the classes of different mutations. We did a sensitivity analysis where we looked at different ways of looking at dose, including cumulative radiation dose, truncated at 1,000 per milligray and so forth, and again, did not find any effect of radiation dose on number of de novo mutations observed. We also looked at birth year of children, and so this is on the x-axis, increasing birth year. We did not find an association with the number of de novo mutations or any of the different classes that I described earlier. We also looked at the nucleotide substitution distribution among the different published at the time de novo mutation manuscripts. Mostly these were the ones that were from the groups that were doing the characterization of de novo mutations in humans, and we didn't find, Orange, our Chernobyl's study, we did not find any different in the class of substitution observed. So a little bit about limitations here. So we actually we weren't able to just evaluate children that were born immediately or conceived immediately after the accident. We looked at a range of years. Although we didn't see an association with birth year, there could be some sort of unobserved survivor bias among children that recruited as adults. We were, for obvious reasons, unable to investigate a lot of acute high doses. And of course, we had an ascertainment bias excluding people that were very highly exposed or deceased. So in conclusion, we found no association observed by radiation dose in any feature of de novo mutations, including the different classes and the numbers of them. We, as I mentioned, the known association with father's age replicated, so our results are very accurate. And we believe that these results should provide reassurance to individuals living in the greater region of Ukraine at the time of accident, because since the people that were enlisted to come in and clean up, their children had no observed effect, that we believe it could be translated into a wider area around this or any other accident. And as mentioned, this was published a couple of years ago, and we actually, I should mention that we have extended the study because we cut the recruitment date. We set a limit on the recruitment date for the original analysis. We're actually expanded it to include more individuals, and we are also going to use the long-read sequencing technologies to address some of the limitations that were mentioned previously. And with that, I would like to thank my collaborators and mention that we are at the NCI very much looking forward to advancing a similar analysis with our colleagues at the RERF.
Thank you, Dr. Eager, so for very interesting observations from Chernobyl children. So we now have 22 minutes for question and answer session. I'm sure some of you may have questions on the presentations, and please introduce yourself and ask questions using the microphones provided.
I want to ask to this Chernobyl data. When you say the normal mutations, there are various types, SNVs and indels and others. When you plot in the vertical axis, you just sum up all those variations?
Yes, so the plots that I showed were of the published de novo mutations in normal populations. And so we just plotted we summed up the number of the different nucleotide substitutions across all of the children in Chernobyl. I should mention that we did do a regression analysis to see, among other things, whether or not there was any class of substitution that was enriched based on radiation dose, and we did not find any association. I should also mention that those nucleotide substitutions, the distribution of them are almost precisely the same when you look across actually human polymorphism. So that gave us double reassurance that what we were seeing was just normal variation.
What's the highest dose you measured?
We had one individual that was over 4 gray. And then the others, there might have been a couple at 2 to 3 gray, and most of the other ones are about a half or.
Thank you very much.
Thank you. Any other questions? For any other speakers as well? You have? Yes.
So related question. How about the thyroid cancer, the mutation of thyroid cancer related to thyroid cancer? We already observed that one. So why do you see anything about that? Oh, Chernobyl.
Oh. Yeah, so if I understand the question is why didn't I mention the thyroid? So the thyroid study, we published back-to-back with the paper in Science. We published a paper about childhood thyroid cancer. So in a similar population, children that lived near Chernobyl in Ukraine at the time of the accident were followed over time, and some of them developed thyroid cancer at a very early age. And so these are individuals that had already been born, or at least were in utero at the time of the Chernobyl accident. So it's a very different study, and those would be akin to the somatic mutations that were described, where we do know that there are, it seems to be that germ cells maybe are more resistant than somatic cells. And so we did see in the thyroid study an increase in the number of indels, just like had been shown. But what we were looking at in the study that I described was specifically whether or not there was a transgenerational effect of children conceived after the accident.
Yeah, at the back.
Thank you. Thank you for your presentations. I have a question. So I think there's not much F1 effect seen in human cohort, but like Dr. Chimura's studies, there are clear effect for the mice studies. So do you think any specific reasons, or like do you think you might see effect in next generations, or maybe maybe like keep tracking population like for more than like F1, like like my studies, or do you think it's unlikely to see next generation's effect in human population? And then if you have any possible reasons, please, yeah.
Okay, so the mouse studies and the human studies had very different exposure profiles, right? So the people that were brought in to clean up after Chernobyl were only exposed for a few days, of weeks, or I mean, I guess some of the people that lived in the area were longer, whereas the mouse studies are more extensive and the exposures happen for longer periods of time. And I imagine that if a situation like that could arise in human populations, that we might see the same thing, but we didn't see. I think the answer, or the way that we're looking at it is that the sort of acute human exposures might not be as detrimental as the longer-term exposures at high dose in the rodents.
Yeah, one more thing.
Mike Weil from Colorado State. A question for Dr. Gondo. You mentioned infertility. I believe it was the high-dose group at G2, and I was wondering how much dose they had accumulated at that time. And a second question is, which strain did you use? Was this an F1 hybrid?
Yes, thank you for your question. All the mice are C57 Black 6J, and exactly the same as Dr. Uchimura's. And B6 Black 6J may have a little higher background mutation rate compared to wild type, because we've done spontaneous mutation study on MSM, which derived from Japanese wild mice. That shows 20% degrees of spontaneous mutation rate. So obviously, even in the same mice, the mutation rate, spontaneous mutation rate per se, were quite different from strain to strain, I guess, because it's just the beginning of the studies, so we don't know yet completely. And your question was?
Accumulated dose for infertility.
The date mated was August 25th, 2021.
No.
Then G1 mice, G1 pair was mated late August. Then kept mating, and G2, we kept producing about a year, but none of the females become fertile. That's the result. So see, G1 mice exposed at the age of 8 weeks, or maybe quarantine, so maybe 3 months old. Then mated, then they kept producing G3, males and females, but none of females were fertile. That's the result. Is it good enough? Total dose, 20 milligray. So earliest.
So it was never more than 20 milligray.
Oh, rate was 20 milligray consistently per day.
Per day. Okay.
Per day.
Okay. And to a total dose of?
Say earliest were born like 1 month later, so you can multiply.
No, just for the individual mouse, it became infertile.
Which one are you talking about?
I'm talking about any one of the generations. How much total dose did those mice accumulate before they were mated?
Well, they were kept irradiated at the consistent rate, 0.05, 1, 20, from the mating time of G1 pairs.
Let me ask a slightly different way. 20 milligray per day for how many days?
The longest is about 360. I'm sorry, 300 days, about 300 days.
Got it.
But that was made to control females.
Thank you.
Well, it's easy to explain if we can have a pedigree here. But anyway, none of the G2 became fertile.
That's one of the reasons the mouse, female mouse, is not a good experiment for the human study.
Correct. The LD50 for resting oocytes is about 0.15 gray.
Any other questions? Yeah. We have some time.
I want to ask to Chimura-san, when you say the Inder, is there, I mean, do you have some profile of the Inder distribution as a function of the number of vacancy? I mean, just take for the deletion case, then just one vacancy, two vacancy as a function of that, how it distributes. Can you have such a profile?
Okay, yeah. Main of the deletion observed in the irradiated somatic cells and germ cells is mainly within 20 base lengths. And most of them, half of them have a microhomologous region at the end of the deleted site. I think this is a very specific signature of the radiation-induced mutation.
My intention, my question is, if profile just of Inder is just going down, then we can just identify this is the number of Inders. But if this is still just it abruptly stopped, then there should be still more Inders you have to watch. This kind of question.
I can understand, but a little bit difficult to answer precisely because short read sequencing technique is a little bit large size deletions are missing, frequently missing. So it's very difficult to answer.
So that means when you say Inder, it's still a part of the entire Inder DNN.
Yeah.
Thank you very much.
Yeah, I have a question to Dr. Noda. So you mentioned that in atom children there are studies going on. So the whole genome sequencing or next generation sequencing studies are planned or are now undergoing in the cohort, or some of them have been already finished?
We are still planning the final stage of the planning. So we will ask the participant consent, because we have already the old informed consent for the study of the genetic effects to ask them the blood donation. But this time, this is a whole genome sequencing. We need another new informed consent. So we will start soon to collecting the informed consent. And then once the number of the informed consent is enough, we will start real analysis.
So how about the archive samples? What are the plans for that, getting ethical approvals, for example?
Yeah, we're not sure how long it takes, but maybe totally 2.5 years or something.
No. For the archived samples, you need to get again approval, ethical approval for. Are you planning to do next generation sequencing
Yes.
on the archived samples as well?
Sure.
Yeah.
Yes.
So that needs again approval.
Yes,
Okay.
because it's in line of all the informed consent, but we need to be sure that ask them for genome analysis.
Okay. Thank you.
Any questions, comments? So after hearing all presentations, the only positive data for the induction of transgenerational effect at the DNA level is still the offspring whose father or mother are exposed more than 3 gray or so. So I'm a little bit wonder, Dr. Uchimura, you are going to examine 290 F1 whose father or mother had been exposed to more than 1 gray. But still, it might be difficult to, find the increase of the mutation, isn't it?
Yeah, yes, thank you. It's a very good question. Yeah, of course, the result is nobody knows, but we try to do our best effort to detect any type of mutations and to compare in such the high dose group and control group, and if there is some possible evidence, we like to find such evidence very clearly, but just to do our best effort. Sorry, thank you.
So how?
We have some earlier statistics expert here in ICRP. So based on the baseline or spontaneous mutation rate of human that published already, and some of the mouse study, we earlier calculated the statistical power to detect the significance. That is the reason why we choose 290 exposed and the same number of controlled. This is based on our calculation.
But such estimation is based on the animal experiment results. So the result I don't know is true. And if that's such same calculation, that such if mouse induced level is observed in human data, Dr. Iga's experiment also detected some tendency of the mutation increase. So I think that a little bit different from the mouse experiment and human experiment.
Even in the same human experiment, not experiment, but human research. Sorry, no, forget it. And there is a discrepancy between the mini-satellite instability between Chernobyl and A-bomb survivors. Do you have some idea why it occurs? Why this kind of discrepancy happened?
Yeah. Thank you. Very, very good and very interesting point. And our whole genome sequencing analysis, usually we use such analysis that we cannot detect most of the mini-satellite mutation. So such mini-satellite region mutation are not have not been assessed using the latest technique in Chernobyl and also in ICRP. So that's a very important future issues in this field, I think.
I have a different question to Dr. Uchimura. So you in the conclusion, you mentioned that germ cells are more resistant to mutation induction. So can you just clarify that more? Because we need to if this is published somewhere, then everyone believes that there is no effect of radiation on the germ cells. But classical radiation biology tells us that proliferative organs and tissues are sensitive to ionizing radiation exposure. So are you including stem cells in the germ cell population, or you're excluding stem cells in the germ cell population? Because at the same time you mentioned that stem cells are prone to develop mutations after exposure. So can you just clarify on that, please?
Yeah. So such proliferation rate is in spermatogonial cells and hematopoietic cell. How fast is I don't know such precise data, but I think I guess germline proliferation rate is not so high, at least a constant state. So possibly this affects such number of the induced the number of mutation and induced by radiation is differ from the such difference, and possibly the human proliferation rate in spermatogonia is much slower. So possibly the such proliferation rate in spermatogonia cell is a very important factor, I think.
Okay, you want to summarize? Okay, any more questions? We have 30 more seconds left for these sessions. Anyone? Last minute question? If there are no questions, we should thank all the speakers for accepting to speak here, as well as for their great presentation. Thank you very much. And to the audience as well, sitting here for the whole session, you're wonderful. Thank you very much.
Thank you.